Are glaciers an archive for 15,000-year-old microbial life? Stay tuned for a discussion of a research study that drilled to the depths of ice to find out. Hey there, welcome to Science is You. We're a growing channel dedicated to making real scientific research available, accessible, and fun for everyone. To support this mission, please subscribe and spread the word. Thanks for watching. So why even look at microbes present in glaciers in the first place? The microbes present in the core of glaciers can potentially represent what microbes were in the atmosphere and landed on the glacier's surface prior to freezing over. So drilling into the glacier core is like going back through time to investigate microbial communities that were present millennia ago, giving us insight into past microbial life on Earth and also environmental conditions that might have affected these ancient microbial communities. Microbes in glaciers were first investigated in the 1980s. There are around 100 to 10,000 microbes per milliliter of glacier ice. Although this sounds like a lot, it's still far lower than other places like seawater, where there are 1 million to 1 billion microbes per milliliter. The cryosphere refers to the frozen regions of the world, or all of the water that is in solid form like snow and ice. Deep freezing is a wonderful way to preserve life, so it makes sense to study the cryosphere to get an idea of past and even ancient microbial life on Earth. Bacteria can also exist as spores, which further enables their survival in extreme environments. Bacteria have even been cultured from ancient glacier ice, including some believed to have been preserved for over 750,000 years. A common way to study microbes, though, is by sequencing their nucleic acids, DNA and RNA. This is called metagenomics. In metagenomics, a sample consisting of nucleic acids from unknown organisms is sequenced and then traced back to try to identify what species are present, sort of like comparing fingerprints. Metagenomics is commonly used to study microbes present in environmental samples like soil, water, feces, or ice. Research has shown that viruses are abundant in the cryosphere, but much less is known about them. In general, microbes and especially viruses trapped in glaciers are difficult to study because they're not very abundant or have low biomass. And also a real challenge is contamination during the collection and sample processing from external sources present on the surface of the ice. The researchers here employed methods used for seawater to sequence tiny amounts of viral and microbial nucleic acids, and developed procedures to make sure the ice samples were completely clean and decontaminated. Here's how they did it. The researchers took 8 samples from the core at a glacier drilled at several different depths, from 13 meters up to 50 meters deep, at the summit and plateau of the Glacier ice cap in northwestern Tibet. This would correspond to a time range from around 70 years ago to over 14,000 years ago. When they drilled out ice cores, no special decontamination was used during drilling. So after obtaining pieces of ice, it was critical that they first decontaminate the samples. But how could they know if the samples had been adequately decontaminated? If we're researching unknown microbial communities and we don't know in advance what we're looking for, it's virtually impossible to tell whether we're looking at our ancient microbial sample or present-day contaminating microbes. The researchers here came up with a creative way to optimize a decontamination procedure prior to handling their samples. The researchers took artificial ice cores and coated them with a laboratory strain of bacteria, a laboratory virus, and a viral DNA of known sequence to simulate contamination from various microbial sources. They then performed 3 sequential steps consisting of washing and cutting to remove 1.5 centimeters from the outside of the ice core, leaving a clean inner ice core sample. After decontaminating and cutting out the inner center of their artificial test samples, they checked for the presence of contaminating microbes using PCR, or polymerase chain reaction, a sensitive technique to detect nucleic acids. They reported that these methods left clean inner ice free of detectable levels of contaminants. Now, with a working protocol to decontaminate their ice samples of any present-day microbes lingering on the outer surface of the ice, they were ready to look at their actual samples of ancient microbes. So they isolated and sequenced microbial nucleic acids in the ice samples. They compared microbial samples taken at the summit and plateau. They found collectively over 254 bacterial genera. Genera refer to species that are classified together based on similarities, like major categories of bacteria. Perhaps not surprisingly, many of these bacteria were psychrophilic, which means they are suited to survive in cold climates. The plateau and summit samples shared some genre of abundant bacteria, including flavobacteria, which are present in soil and can cause disease in fish. But several types of bacteria that were present at the summit were interestingly not present at the plateau, such as methylobacteria, often present among plants. Methylobacteria were abundant in ice drilled from the summit. This showed that there are potential differences in ancient microbial communities present at the summit and plateau of this glacier. Using a data analysis method called principal coordinates analysis, a method to look at how similar or different two sets of data are, they found that the samples from the summit and plateau overall consisted of different microbial populations. The authors then looked at viral genomes from two samples taken from the summit. From the shallower and deeper depths, which would represent a few hundred years ago, and over 14,000 years ago, they were able to group the sequences into 33 likely virus species. Of these, they were able to identify only 4. The rest were unclassified. The 4 identified were viruses that infect bacteria and other microorganisms called bacteriophages. This indicates that glacial ice has a diverse and unique viral community. The authors point out that the percentage of unique, unclassified viruses in glacial ice is much higher than in other environments like oceans and soil. They then compared their dataset to that of viral datasets taken from other environments, and found that other samples taken from lakes, soils, deserts, and other ice sheets didn't contain any of the 33 virus species sequenced in glacier ice. This indicates that glacier ice has a unique viral composition as compared to other environments, at least based on what was detected in this study. When they compared viruses taken from the different depths of the ice, they found that there were some viral genomes in common between the two samples, and some that were different. The authors suggested that the glacier could serve as an archive for microbes that were adapted to survive in colder temperatures, that could perhaps become active again if the glacier were to melt. The authors further speculated, based on their nucleic acid sequences, that the bacteriophages they found may have been infectious to the bacteria present in the glaciers, forming a dynamic microbial community. Still, a limitation of this study is that the authors could have missed some of the viral and microbial diversity if some species were below their limit of detection. Also, there isn't that much known about viral communities in glaciers, but what the researchers did find with their development of decontamination procedures may have unlocked opportunities for finding out more. What do you think? What can the microbial populations of glaciers tell us about Earth's past ecosystem, and what might it mean for the future with climatic changes? Share in the comments. If you liked this video, please subscribe and share to help this channel out, and remember,